Today in Chapter 6, we're going to cover DNA and DNA technology. Now, the scientist you see here is Rosalind Franklin. She's born in 1920 in London, England, and died in London in 1958. Now, she's going to have remarkable scholastic aptitude. She excels in science from a very young age, and she's also really, really good with math. She's one of those who, when she was in school, she always expected to be the best at what she did, and she made sure that she put in all the effort and all the time to make sure that she was the best. Now, she attends the Newnham College in Cambridge and earned her degree in 1941, and then went on to the University of Cambridge for her doctoral degree, which she received in 1945. She's going to work with British coal utilization research during World War II, and it actually led to the development of a better new gas mask that they could use during the war. She later on started working with crystallography, X-ray crystallography, and that's also known as diffraction, and she did this at King's College in 1951. And what she was doing is she was studying the DNA molecule. She then went to Birkbeck College in 1953 to study genetic material and structure of viruses. And that's where she was working when she passed away. So she is known for the famous Photo 51. This photo she took while she was at King's College, and it was a picture of DNA. So when she's working with her crystallography, she was taking pictures of DNA strands, which was very hard at the time because DNA strands are microscopic. And so as she's taking these pictures of the different structures, she came across one picture that she developed, and she kept it to the side. And this is going to be the picture that helps her figure out that DNA is going to be a double-stranded helix. And it also helped her to figure out how it was matched up. And so that photo got the name Photo 51, and that's how it became famous. Now, the name is going to appear in multiple building awards, and as well as an asteroid even. And so we have the asteroid 9241, and that's the Rosalind Franklin asteroid. And that was discovered and named in 1997. Now, some other scientists that help with discoveries of structures of DNA, we've got Frederick Miescher, that is in 1860. So what he discovered is going to be the nucleon. That's going to be an unknown substance with a unique chemical structure in the nuclei of white blood cells. This is what he believed influenced inheritance and traits. Next, we have Albrecht Kossel. That is in 1868. He died in 1901. He isolated nucleic acid and identified the 5 nitrogenous bases: adenine, guanine, cytosine, thymine, and uracil. We have Fabius Levene in 1919. He isolated and identified the basic components of DNA, including its nitrogenous bases, which we said, the adenine, cytosine, thymine, and guanine. And he discovered that the sugar that's going to help to make up the DNA molecule was deoxyribose. He also found that the phosphate group is going to be linked with that sugar in, and then those help to keep those nucleotides together. Then we have Nikolai Koltsov in 1927. He proposed the idea that the inheritance of traits occurred by means of large molecules that replicate in a semi-conservative fashion, with each strand acting as a template. We have Frederick Griffith, 1928. He's going to demonstrate unidentified transforming factors are going to be responsible for the transfer of traits from 2 forms of pneumococcus. We have Jean Brackett in 1933. He's going to discover that DNA is going to be located within a chromosome of cells. And so the chromosomes, you should know, are also going to be found in that nucleus. William Atsbury in 1937 is going to produce the first X-ray diffraction pattern of DNA molecules, which he suggested could be used to easily deduce its structure. We have Oswald Avery, which is in the first picture. We have Colin McLeod, which is going to be in your second picture. And then Macklin McCarty, and that's in your third picture below. In 1943, they're going to discover that DNA is going to be the transforming factor resulting in the transfer of traits using that pneumococcus bacteria in that Frederick Griffith's 1928 experiment. And so the experiment was done first with Griffith, and then it was done again, and then these three scientists were able to prove it. So what happened is they're going to take 2 strands of pneumococcus bacteria. One's going to be a virulent strand that causes pneumonia. The other one is going to be a non-virulent strand that does not cause you to become ill. They find out that if they take the bacteria that's non-virulent, does not cause illness, and they expose it to bacteria or cells from the one that is virulent, the non-virulent cells will pick up the strands of DNA from the virulent bacteria, and now the ones that did not used to cause you to become ill will now cause you to become ill. And so something had to be inside of those bacteria that allowed this to happen. And they called that, or they discovered and named it DNA. Then we have Alfred Hershey and Martha Chase in 1952. They're going to conclude from the bacterial phage experiments that DNA is going to be genetic material. Bacteriophages are going to be viruses that are able to infect bacteria. And so these viruses are very, very small. Bacteria are extremely small, but the viruses are even smaller. So you can have hundreds of these viruses on a bacterial cell. So let's get into this DNA structure we keep talking about. DNA is going to be made up of subunits or units called nucleotides. Those nucleotides consist of a 5-carbon sugar. It could be ribose or deoxyribose. It could be made up of a phosphate group that's hooking on to that deoxyribose in the case of DNA, and then you'll have your nitrogenous bases. And so you can see that phosphate sugar on the side. That's going to make up what we call the backbone of DNA, or the sides of the ladder. So hold on just a minute. And so these sections right here that you see in like this silvery blue, that's going to be made up of the phosphate sugars. And so they'll alternate phosphate sugar, phosphate sugar, phosphate sugar. Now attached in with that sugar is going to be what we refer to as the nitrogenous bases. And so that's going to be your A, C, T, and G's. And so these are going to be the different colored portions that you see here. Now with those different colored portions, they're going to be hooked together in the center with a weak hydrogen bond. And so the sides that we saw here, the sugar phosphates, they're going to be a very strong bond. So they will not come apart. It's going to be important to keep those nucleotides or the nucleic acids in a straight line. You don't want them rearranging themselves. And so that's going to be a very, very strong bond. The center, a very weak bond, and it's a hydrogen bond. And that's going to be important because DNA is going to have to unzip itself, open itself up, make copies of specific genes, specific proteins, or make copies of itself in its entirety if it's going to go through mitosis or some type of cellular division. And so it has to be able to open back up and then close back up to reform its structure that it had. So here you see it contains 2 alternating covalent bonded sugars and phosphate molecules, and that's the backbone. Remember, the backbone is strong. This structure is going to be deduced by the scientists Watson and Crick, and that's going to be in 1953. And was allegedly based on mathematical data collected by Rosalind Franklin. And we say allegedly because we're going to view a video, the photo 51, and it seems that they were able to sneak into her lab when she wasn't there in the evenings, and they were able to get a hold of her research. So they were the ones who created the model and won the Nobel Prize for it, but they used her research in order to create that model. And when they did that, she did not know, but she was the main contributor of that data. Right, so with this arrangement, you have your strands. They're going to run anti-parallel. So it's an anti-parallel arrangement. So that's going to result from one strand that ends in a 3 non-binding sugar and the other in a 5 non-binding phosphate. And so they run in opposite directions. So numbers reference in the carbon in which the model or the molecule is going to be bonded. So you have one strand that runs 5 to 3, the other strand runs 3 to 5. And so let's say one strand, get my pencil, one strand could run down, one strand will run up. And that's what it means. It's going to run anti-parallel or opposite each other. So with the nitrogenous bases, orientation first is going to be suggested by Rosalind Franklin, because she's going to be able to look at her pictures like photo 51 very clearly and deduce how they're arranged together. It's going to be located in the center of the DNA backbones. So it's going to spell out the genetic code that's responsible for building a living organism. And we're going to have like two types. We have what's called pyrimidines, and that's going to be a single ring structure that consists of thymine and cytosine. And you'll have purines. Those are going to be double ring structures that consist of adenine and guanine. And so if you look at those, let me circle those for you. And so if you're going to have your singles, you'll notice this is going to be a single, and that's your cytosine, and this one's going to be a single, and that's going to be your thymine. And then your doubles are your purines, and so we have our adenine here, and we have our guanine here. And so notice a single and a double will always match up. Now with these, if it's adenine thymine, it's going to hold together with a 2-strand hydrogen bond. If it's guanine and cytosine, it's going to have 3 hydrogen bonds that's holding them together. We're going to have what's referred to as Chargaff's rule. This is going to be introduced by the scientist Erwin Chargaff. It's going to state that the number of adenines are going to equal the same as thymines, and while the number of guanines is going to equal the same as cytosines. And so they do this by analyzing the data and analyzing the nitrogenous bases that they come up with when they run their analysis. And so that means for every adenine, it's going to have to bind with a thymine, and vice versa, and every guanine is going to have to bind with a cytosine. And that is your Chargaff's rule. Now, organization of organisms, you're going to have your genome. That's going to be the organism's entire set of DNA. Bacterial genomes consist of a single circular piece of DNA that's going to be attached to the plasma membrane called a nucleoid. And so it's kind of just floating around in the cytoplasm, but it does have one set attachment point to that membrane. Eukaryotic genomes are going to be found within the nucleus of the cell. That's going to be where DNA is going to be wrapped around these proteins that are called histones. Those proteins keep it from tangling up, and it will also enable it to be tightly packed in there with that nucleus. The human genome is going to contain 23 pairs of chromosomes with over 3 million base pairs. And so 23 pairs equals 46 total chromosomes. So when we say pairs, make sure that you're paying attention. Is it saying chromosomes or is it saying pairs of chromosomes? If it says pairs, you'll have 23. If it's chromosomes total, you should have 46. Great. Insect-dwelling bacteria. These are going to have the smallest genome. They're only going to have about 112,000 base pairs. The organismal genomes, things such as dogs, are going to have 39 pairs of chromosomes, where fruit flies will only have 4 pairs of chromosomes. So the length of DNA molecules is actually going to be determined by the number of base pairs in an organism's genome. So the number of A, C's, T's, and G's that are there. A gene is going to be a unique sequence of around 3,000 or up to 3,000 DNA bases. Genes are going to code for specific things. They'll code for synthesizing proteins. They're going to be located on specific areas on the chromosome, and that's going to be first discovered by Thomas Hunt Morgan in 1910. So those specific sections of the chromosome are going to be referred to as the locus. So let's say we have a gene that is going to be found within this section here of this chromosome. That would be its locus. It's found in multiple locations is referred to as loci. So you get LOCUS for a location. Sorry about that. Let's exit drawing and see if it goes back down. I apologize. And so LOCI, loci, That's going to be it's located in multiple sections of a chromosome, or maybe on multiple chromosomes. And there are quite a few genes that aren't just located on one chromosome or in one section. They're located in multiple different spots. And so you put those areas together and that gives you the gene. Notice here also, you've got your histone proteins. These are going to be those proteins that the DNA wraps around. Notice how it can keep it nice and untangled, and it also helps it to condense itself. Now, during normal interphase, that's a normal cell life, it's going to be found in the nucleus in a stringy form. That stringy form is called chromatin. But when it's getting ready to divide, it will condense and it becomes very tightly coiled and makes this chromosome shape. With DNA, you'll have certain sections that code for proteins and you'll have certain sections that do not. Non-coding DNA does not code for proteins, and it can exist as what's called a gene fragment. It can be a duplicate of a gene, or it could be a mutated gene. About 25% of non-coding DNA is going to be found within genes, and it's known as introns. So where you see it not lit up, but it's found within that structure. So let's say our gene covers this entire area right here. This section that's gray in the center is your intron, and so those will be your A's, T's, C's, and G's that don't code for a gene. So those are going to have to be taken out before that gene is sent to the ribosome to be made. So with these, the introns are going to be discovered in 1977, but the term's not actually going to be used until 1978. So between 65 and 100,000 DNA bases are going to be found in an intron. So that's a lot of A's, T's, C's, and G's that are going to be found there. And they don't code for anything really, or at least they don't think so. So the function is not fully known. However, they say some scientists suggest they may be involved in regulation of mRNA formation during translation. They could be part of crossing over during meiosis, or part of switching genes on and off, which influences protein synthesis. I've also seen where research kind of suggests that those could be what make up our unique personalities, but they haven't quite found that connection yet. So the remaining 75% of non-coding DNA is going to be found between these genes. So you might have that on these sections, and you might have that on these sections. Exons are going to be the areas that code for proteins. So that's found within the gene. Usually it's anywhere from 100 to 300 bases, and this is going to code for whatever specific thing that you need in that protein. Let's say you are getting ready to eat some ice cream. I always use ice cream or milk, and you need some lactase. So you will go in, mRNA will copy the gene for lactase. It copies this entire section here, and then it's going to remove the introns and then hook the exons together, send that to the ribosome, and then make that lactase. It'll then send it to the intestines, the small intestine, which will allow you to break that sugar molecule down, and it'll be then in a usable form that your body can absorb and use for nutrients and energy. So cell division, that's going to be an important process that a cell undergoes. Prior to division, a cell has to synthesize or create more DNA, more organelles, more cytoplasm. So it has that cellular material that's in there that it's starting to duplicate. It has to have more nucleotides in order to double up the DNA. It's going to have to generate energy before DNA replication can occur. So it has to proceed with a cell that's getting ready to divide by duplicating or replicating its DNA. So that DNA will open up, comes in, the two original strands that you have there that broke at those hydrogen bonds are then going to be copied. And so then you'll end up with two daughter cells that have the exact same DNA and the exact same genetic material. So here's what we're going to do. This is called the conservative model. It's going to state that the parental DNA strand was preserved and comes back together once replication ends. And so you have this suggestion here, and there's two different models, or three different models that are suggested. So you see how it splits through the center. That's where your hydrogen bonds were. And you're going to have two different strands. They come back together here. And then you had two new strands. They came together. So that's what's referred to as the conservative model. You preserve the parent strand, and you have two new daughter strands that come together. Then you have what's called the dispersive strand or model. With that one, these strands are going to come together. They're going to consist of a mixture of both the new and the old. And so you'll see the light blue is the new, the dark black is going to be the old, and it looks like it's kind of intertwined with each other. Then lastly, you'll have what's called the semi-conservative model. And this is the model that they actually have discovered is going to be the model that actually happens. So it's going to state that replicated DNA strands contain one parent strand and one newly formed strand. The idea is first going to be proposed by Koltsov in 1927, but officially is proposed by Watson and Crick in 1953. Through experiments performed by Matthew Michelson, getting tongue-tied today, and Franklin Stahl in 1958, that's going to support the semi-conservative model. And so it disproves the other two. So what we end up going with, what is the correct model? It is going to be the semi-conservative model. So with the DNA replication, you're going to have a starting point where DNA is going to open up and kind of unzip. That's going to be referred to as the origin of replication. It's going to be a short, specific region of DNA where it's going to start to break apart. You have an enzyme called helicase. It's going to recognize and attach to the origin of replication, and it begins to unwind and separate that DNA molecule into two parent cells, or parent strands. So it goes in and it does that by breaking those hydrogen bonds that are holding those nitrogenous bases together. And so looking at it, you see how your DNA helicase is here. It, on this end, has already broken those strands apart. As they break apart, it's going to break those hydrogen bonds, separates them. Nucleotides that are floating around in the nucleoplasm are going to come in and bind with their complement. So let's say one of them's an A, well, a T is going to come in and bind if it's making another strand of DNA. Now, DNA ligase is going to help to bring this in and attach these, and then you have your DNA polymerase, which kind of reseals those hydrogen bonds and puts it back together for you. Now, right here where you see it coming in and breaking those bonds, that is often referred to as a replication fork. And so it occurs at that replication fork where those hydrogen bonds are broken, kind of like a fork in the road or a Y. That's where it's breaking. And so it breaks it apart and makes single strand binding proteins. They're going to come in, attach those parent cells together, and help keep those strands separated. So RNA primer, that's going to be a short sequence of nucleotides. It's going to be produced by the enzyme DNA primase. RNA primer is going to come in, it attaches to the DNA polymerase. That's an enzyme responsible for complementary base pairing, the correct nitrogenous base to the exposed base on the template strand. Using a base pairing rule, A always forms with T and G always forms with C. So it's going to come in and it makes sure that it primes properly. DNA replication is going to occur in that anti-parallel direction. So you're going to have part of it occurring going one direction and a part occurring going the other direction. Lagging strand is going to be the new strand that's being created by complementary base pairing from 5 to 3. It discontinuously is going to be created by small fragments called Okazaki fragments, and that's going to contain around 100 to 2,000 nucleotides depending on cell type. Okazaki fragments are then going to be joined by that DNA ligase, and so that's going to lead to a strand that's going to be new and is created by that complementary base pairing from that 3 to 5. Now, leading and lagging strands are going to replicate in both directions at the same time. So you're going to make these anti-parallel strands both at the same time, matching up with the original parent strand and making that complementary. Elongation rate of new DNA strands in human cells is going to be about 50 nucleotides per second. So that's how quickly it is building this DNA. 50 nucleotides per second. So each new strand is going to be complementarily pairing its bases. Then it's going to be proofread by different types of DNA polymerases. The reason it has to go through and proofread is because sometimes it might accidentally match up the wrong base. You might have an A and a G matched up, or a T and a C matched up. And so if it finds an error like that during its proofreading, it's going to use an enzyme called nuclease, which is going to cut out that wrong fragment of DNA, the wrong base, and it's going to take, and it uses DNA polymerase and DNA ligase to fix it and bring in the correct pairing. So if you had the A and G together, it will then cut that G out and bring in the T and make sure that it's correctly paired and fix that strand. So DNA replication is going to end when DNA polymerase removes the RNA primer and then adds the correct DNA nucleotides to the RNA region. Next, we're going to cap the ends of the chromosomes. These caps are referred to as telomeres. So they're short sequences of repeated nucleotides that lack genes, and they don't code for any proteins. They're actually going to kind of act as an odometer. They're going to count how many times that cell is divided and it's replicated its DNA. So its function is to count the cellular divisions, and it's going to help to protect the ends of DNA. So it kind of acts as a protective cap. Think of the ends of your shoelaces. It's kind of got that little protective cap on that end. Now with this, they've actually been able to go in and they can look at DNA and they could tell with some people if they have had a rough life. You can go in, and if they had a rough childhood, lots of times the telomeres have become damaged. And that allows then the DNA to become damaged. Some people actually are born with shorter telomeres, and so they age a lot faster, and they tend to have more age-related genetic problems and health problems. Some people have abnormally long telomeres, and with those telomeres, it's extra protection, and those people tend to have less health conditions, and they tend to look younger for longer. So that's a fun little fact about telomeres. With gene expression, that's also going to be known as protein synthesis. Synthesis means creation or making. So it's going to be directed by a sequence of nitrogenous bases in the DNA. They only provide the code for protein synthesis. They cannot initiate it. And this is going to occur at the ribosome that's located in the cytoplasm. So it could be a free-floating ribosome, or it could be one that's attached to the endoplasmic reticulum. RNA stands for ribonucleic acid, and that's going to be the molecule that helps to decipher the code that's going to be outlined by the DNA sequence of nitrogenous bases. And so the sequence hypothesis is first introduced and described by Francis Crick in 1958, and it's going to state that a piece of nucleic acid is expressed by bases and the sequence of codes of amino acids for a protein. So the central dogma of molecular biology explains that the relationship between DNA, RNA, and proteins is going to be found here. So what happens is we're going to have the replication of DNA. So it's going to open up to a specific area for a gene that you need to copy. It's going to go in and it copies and makes a complementary strand. But this strand is not going to be DNA, it will be RNA. And that RNA is going to be specifically mRNA. And so that stands for messenger RNA. So it copies that strand, but with RNA, remember, you're no longer going to have T's, now you're going to have U's. So everywhere that you should have put a T, now you're going to have a U. Once it copies, that RNA strand is going to then send it to the ribosome, which is going to put it together one amino acid at a time. So each one of those little circles is an amino acid. And then in between, those circles or amino acids are hooked together by a bond, and that's going to be a peptide bond. So at the end of putting the strand of amino acids together, you end up with a polypeptide chain. So with RNA, it's, like we said, going to exist as a single strand. Remember, DNA was double. RNA is going to contain the sugar ribose. DNA had deoxyribose. The nitrogenous base, thymine, is replaced with uracil, and it's going to function as an enzyme. Remember, enzymes are types of proteins. So there's going to be 3 different types of RNA that's going to be responsible for converting the DNA-based sequence into actual proteins. The first one you're going to have is going to be messenger RNA, or often referred to as mRNA. So the M is lowercase, and of course RNA is going to be capital. That's going to decipher the DNA code that's used to make a protein, and it's going to carry that message from the nucleus to the ribosome. Remember, it's going to copy the nucleus, or copy the DNA strand in the nucleus, exit through the nuclear pore into the cytoplasm. That's where it's going to meet up with the ribosome in the cytoplasm. The ribosomal RNA, or rRNA, is going to be the major component of a ribosome that makes up nearly 60%. It's going to be in 2 parts. It'll come together, and it will start to read the mRNA strand. At that mRNA strand, as it's reading it, you're going to have tRNA, or what's referred to as transfer RNA. That's going to take, it's going to read it 3 codons at a time, or 3 bases at a time, and it's going to translate that mRNA code into a particular sequence that codes for the amino acid. It goes out, it finds the amino acid, brings it back to the ribosome, and it starts to make the protein there. So it brings the amino acid back, it forms a peptide, brings another amino acid in, hooks them together, so then you have your first peptide bond. So with these amino acids, these are going to be molecules that make up proteins. There's going to be 20 different amino acids that can be used to build a protein in living things. And so there are more than 20 amino acids total, but these are the ones that we're going to need to actually make our proteins. Some of these are going to be positively charged. Some will have a negative charge to them. Some are going to be polar uncharged. Some will be special cases. You'll have the hydrophobic ones, and you'll have other types of hydrophobic ones that you see in here. So this is the different types. So with the genetic code, you're going to have a tool that consists of about 64 3-base sequences called codons that are going to specify for a particular amino acid. So you have 64 total ways that you can put these together. Codons are going to be 3 bases at a time. So you have 64 different ways you can put those 3 together at a time. So remember, you have 4 total. A, C, U, and G. Because remember, U, we're talking about uracil, it is RNA. So you're going to take it, and you can rearrange those 4 codons in pairs of 3, 64 different ways. So that's what this is saying. Francis Crick in 1961 is going to be the first to introduce this idea that genetic code is going to consist of triplets that could code for that amino acid. PolyU is going to conduct experiments by Marshall Nierenberg and J. Heinrich and Mathia in 1961, and that's going to be where the first ones are going to demonstrate that these codons could specify amino acids as they observed them at UUU and specified phenolphthalein, or phenolphthalein, lalanine. And then so with that one, that's going to come in, make that specific amino acid. Marshall Nierenberg in 1966 and his team are going to reveal that these codons are responsible for coding for 20 amino acids. Only 61 of the codons will specify for specific amino acids, while there's going to be 3 that remain. These 3 don't code for a specific amino acid. Instead, they are called stop codons. So they tell that tRNA, hey, stop here. We are done making this protein. And so that's what happens during that protein synthesis. So with this, words are used to describe the genetic code. They include Degenerate, that refers to multiple amino acids being specified by more than one codon. You could have universal, which is going to refer to all living organisms using that same mRNA codon to specify for amino acids. And you could use what's called unambiguous, and that's going to refer to each codon specifying for one amino acid. And so here, this is looking at your amino acid chart and how you would read it. So if I had AUG, that's going to be your start codon. You would look first on the side. Let me get my pencil. And you would find A. And so here we are. Next, you would look at the second letter, U. So you know it's going to be in this column here. And then you come over to the side and find G, which is here. So you know it was in this row, first column. And so you bring yourself over here, you found that AUG. AUG is your start codon, and it codes for methionine. Transcription is going to occur in the nucleus of the eukaryotic cell. And so what's going to happen there is it is going in and it is copying the gene. That's what transcription is. It's going to occur in 3 steps in which that DNA is going to be processed and transcribed into mRNA. So we have transcription and translation. First step, transcription. We've got to copy it. Starts with initiation. So it begins with what's known as a gene promoter. That's a non-transcribed region of DNA that contains two short sequences of DNA bases that are TA, TAAA. And so once it comes across this, the transcription factor is going to bind to these areas right here at this site, and transcription factors are going to bind to create protein complexes. That's going to enable that RNA polymerase to then bind to that promoter. It initiates transcription, which is copying of the gene. It indicates the direction of transcription, so it tells it which way to go, and which DNA strand is undergoing transcription, so what side it's actually copying. With this, the RNA polymerase is going to go in. It unwinds the DNA molecule and starts to break those hydrogen bonds between the nitrogenous bases, producing 2 separate strands of DNA. You've got the template strand that's going to contain the necessary information that's required to be transcribed and can produce hundreds of mRNA molecules depending upon the amount of protein that you need. The non-template strand, or the coding strand, is not transcribed, but it may be a template for another gene. So that's the first step. Second step, you have elongation. That's going to occur along the region of a gene known as a gene body. As the RNA polymerase travels down that template strand from the 5 to 3 end, the complementary RNA nucleotide bases are going to be paired up to the exposed DNA nucleotide bases. So they're going to kind of match up and make a complement. So anywhere you had an A, you're going to end up with a U. Anywhere you have a C, you get a G. Anywhere you have a G, you get a C. And anywhere you had a T, you're going to get an A. Upon reaching the adenine nucleotide, RNA polymerase pairs the nitrogenous base uracil. So that's what I just described. After the mRNA strand reaches about 10 nucleotides in length, the strand separates from the DNA template strand. So it doesn't stay there until it finishes. It does start to break away pretty much as soon as it gets going down the strand. Those hydrogen bonds begin to reform between the nucleotides of the original 2 DNA strands, and they'll start to wind themselves back up. Once it finally gets to the end of that strand, it's going to reach what's known as a terminal region, or get a terminal signal. That terminal signal is going to be a specific sequence of DNA nucleotides on that template strand near the end of the gene that say stop, quit copying here, and release. So upon reaching that terminal, that mRNA strand gets released, and it's shortly followed by RNA polymerase. So with termination, you're going to end up with a newly formed mRNA strand that's going to undergo 2 types of modifications. First, it's got to cap it. So remember, it's got to go in and put those telomeres in. So it involves adding a single modified guanine cap at the end, an mRNA strand, and a sequence of adenine nucleotides up to about 200 on the 3 end. And that's going to form a tail. Important process because it's going to help to protect and transport the mRNA while also allowing it to correctly align and bind within the ribosome. It also goes through and it edits the mRNA sequence. So this process involves removing non-coding introns and combining all the protein coding exons together. So you're taking out the snippets of the coding that did not code for any part of that gene. So once the mRNA is edited, it's going to travel out of the nucleus through the nuclear pores to the cytoplasm. And that's where it's going to remain for a few minutes to a few days prior to binding to that ribosome. And so here you go. You see that transcription. It hits that signal that tells it to terminate. So it starts to separate. Once it separates, it's then going to cap it. And so it caps it at both ends, takes out the introns, puts the exons together, and then it sends it out the nuclear pore. That's transcription. That's where you're copying a gene. Now we're going to the second part of protein synthesis. That's going to be translation. So we have to take that gene that we just made a copy of, and we're going to translate it into amino acids, hook them together with peptide bonds, and make the protein. This occurs in the cytoplasm. So the process where mRNA strands are translated, like we said, into the amino acids. This one requires what's called tRNA or transfer RNA. So the molecule responsible for reading that mRNA code and delivering the appropriate amino acid to the ribosome. So it's a three-dimensional molecule that's kind of shaped like a clover leaf, and it contains a sequence of 3 bases that are going to complement that mRNA code. And so it reads the 3, hold on, let me get my pencil. It's going to read the 3 codons on the mRNA strand, and it's going to make complements of these. So those are going to be referred to as anticodons. And so it reads CGG to match up with what was on that original strand of mRNA. The anticodon is going to decipher the mRNA codon, initiating the tRNA to deliver the correct amino acid. And it attaches it to the mRNA using ATP. Now remember that ATP is going to be our energy molecule. About 40 different tRNA molecules with at least 1 for each of the 20 amino acids, suggesting that tRNA molecules can recognize multiple codons at a time. This is also known as the wobble effect. It was observed by Francis Crick in 1966 and is going to be very important because it ensures a functional protein is going to be made. So with translation, that's going to occur in 3 steps. So we had the initiation. That's going to begin where you read the codon AUG. That's methionine or methionine. It's pronounced, I've heard it both ways. So that one is going to be the one you always start with. So it's referred to as your start codon. So if you see in any test or quiz, what is the start codon? It's always going to be AUG. Once translation is initiated, an initiation complex is going to form and bind to the start codon, consisting of small ribosomal units with mRNA binding sites and an initiator tRNA molecule. So the initiation complex properly aligns that ribosome and the mRNA strand. I kind of like to think of it as whether 2 ribosome pieces, they come together, 2 subunits, and then the mRNA strand goes through the center. And it's going to read it 3 at a time. So I refer to that sometimes or as like a ticket reader. If you go to Chuck E. Cheese or somewhere and you win a bunch of tickets on your games, you can send it through that ticket reader machine to count your tickets for you. And so think of each one of those tickets being 3 codons. And so it's going to go through and it reads it 3 at a time. Elongation is going to be building the amino acid sequence of the event, which eventually becomes the protein. So ribosomes are going to travel down the mRNA strand from 5 to 3 direction and translate each mRNA. As the tRNA recognizes that codon, it's going to bring the appropriate amino acid back to the ribosome and release it. And then it moves to the next codon and then the next. And remember, as it's releasing it, it's hooking them together with peptide bonds. So once that amino acid's released, it comes in, and that peptide bond hooks it to the next one that's brought in. That creates your amino acid chain, or sometimes you hear it called the polypeptide chain. And then you hit the last step again, which is going to be termination. That occurs when that ribosome reaches the stop codon. Remember, these don't code for any specific amino acid, so those are going to be UGA, UAG, and UAA. So you might want to memorize these three and know that these are stop codons. If you see these and I ask you what are these codons, they're the stop codons. And so if it comes to this, that mRNA strand starts to terminate that protein synthesis and the ribosomes start to pull away. So releasing factors are going to bind to the ribosome that cause it to kind of pull away and release that mRNA strand. You have amino acid sequences beginning to fold into a functional protein, because remember, once you make the strand, that's just the primary structure of a protein. You still have to put together the secondary, tertiary, and quaternary structure before it becomes functional. So they start to fold, which is going to be your secondary structure. The three-dimensional protein structure is then going to help to chaperone these proteins after being released. So chaperone proteins are going to influence the correct molecular interactions and help to stabilize all this folding and connecting to make that final structure. So proteins may require additional modifications such as amino acid rearrangement or attachment to other types of molecules. And so here, this is just an example showing you translation. So you're going to have your large ribosomal subunits come together. It's going to read these codons 3 at a time. The tRNA, which is that clover leaf, or kind of looks like a T actually, comes in. It's going to make anticodons for those codons, go out, find the amino acid, bring it back, and start hooking them together. As it hooks them together, it's going to create that, or pull them together with that peptide bond in between each one of those. Mutations. These are going to be an alteration of DNA or RNA-based sequences that can affect the overall structure and function of a protein. Typically, they can be harmful, producing non-functional proteins that can lead to serious diseases or death. Non-functional proteins will produce formed mutations such as enzymes that don't catalyze properly, or it can make it so chemical reactions don't occur within the body. So the inability of these enzymes to catalyze chemical reactions does result in a buildup sometimes of those molecules they were supposed to break down, and that would have reacted to the enzyme that would have broken down. So if it builds up, it can become like a toxic environment there. So some mutations, though, could have no detrimental effect, while others could actually be beneficial to an organism and be favored by natural selection. So it could have 3 fates. It could be detrimental, cause problems, disease, death. Nothing could happen, or it could be a benefit. With these mutations, you're going to have different types. Spontaneous mutations are going to be the most common type of mutation, and they come from accidental errors that occur during DNA replication. And so that results in the substitution, insertion, or deletion of base nucleotides. During and after DNA replication, we have DNA polymerase that's going to go through and proofread the newly formed strand of DNA. If it finds an error, that DNA repair enzyme is going to cut it out and repair the incorrect sequence or mistake. So that's going to help to minimize the possibility of spontaneous mutations occurring, and it makes those types of mutations more rare. Induced mutations are going to occur because of toxic environmental factors called mutagens. There's 2 different types. You can have chemical mutagens. They're going to interfere with the atoms in the DNA molecule that affects the ability to replicate. So those could be things like cigarette smoke. Radiation mutagens, those could be like ionizing radiation, things from X-rays and gamma rays. The use of X-rays and how it contributes to mutations is first going to be observed in 1920 by American geneticist Herman Mueller. Exposure is going to affect the atomic structure of DNA, and it helps to break the DNA apart at the chromosomal level. Radiation mutagens are going to be non-ionizing radiation like UV radiation. We'll get that from the sun or from tanning beds. That's going to cause bond rearrangements that could prevent complementary base pairing of pyrimidines. This is going to form kinks within the DNA structure with adjacent thymine bases covalently linking and producing thymine dimers, and that's going to affect DNA replication. So thymine dimers are going to be removed by that DNA repair enzyme. Induced mutations could also have what we call chromosomal aberrations. This is going to be a large-scale mutation that affects the overall organization of a gene, and you can have 4 different types. Inversion is going to occur when the DNA is removed, inverted 180 degrees, and then reinserted into that chromosome. And so it takes it basically, flips it upside down, and reinserts it. An example of that would be hemophilia. That's what happens when you inherit that gene. Translocation is going to involve the removal and relocation of a DNA segment, either into the original chromosome, or it might put it into another chromosome. One of these could be an example of Down syndrome. Deletion can occur when a segment of DNA is going to be removed or lost from a chromosome. An example of that would be like Williams syndrome. So I'm going to try to finish this slide up really quick. The fire alarms are going off, but it's a false alarm, so I'll start a new video once it is finished. Okay, so I want to finish this slide though. Duplication is going to occur when a DNA segment is duplicated either on the original chromosome or a different chromosome, and that could be like CMT disorder.